Hello everyone, welcome back to my channel Anytime Medicine, and today we are going to see is glucose. So before we start, subscribe to my channel so you cannot miss my future videos. So glucose is a carbohydrate and most of them have formula of number of carbon and number of H2O, which is water, and that's why carbohydrate also called water carbon. As you can see in the glucose structure that it has so many hydroxyl group and hydrogen bond in the structure. There are certain groups of carbohydrate. One of them is monosaccharide, which has 6 number of carbon and 6 number of water molecule in it, and those are glucose, fructose and galactose. As you can see in the picture, the glucose, fructose and galactose have same number of carbon and same number of water molecule in the structures. Only the difference is that they have different chemical structures and they are isomers of each other's. Another group of carbohydrate is disaccharide, which have two monosaccharide structures in their structure. They will break down to monosaccharides in GI tract. The first is the lactose, which is made up of galactose and glucose and which is broken down by lactase in the GI tract. Second is sucrose, which is made up of fructose and glucose, which is broken down by sucrase in GI. As we have talked in last slide that lactose is broken down into galactose and glucose by lactase into GI, and sucrose is broken down into fructose and glucose by sucrase in GI. Third group is polysaccharides, and they are polymers of monosaccharides and they are linked to each other in a series or in a complex molecule. So first one is a starch, and a starch is plant polysaccharides and which is glucose polymer. Second is glycogen, and which is animal polysaccharides and which also is a glucose polymer. Third polysaccharides is cellulose, which is also plant polysaccharides which is made up of glucose, and this is different from starch and it cannot be broken down by animals. That's why we give them in diet as fiber for improving the bowel function. Remember that all carbohydrates always break into glucose, fructose or galactose, any of these three structures. Now we'll see how glucose will need in metabolism. Glucose used in anaerobic metabolism and their end product will be lactate, and secondly it will used in TCA cycles for ATP in order to generate energy, and their side product will be water and carbon dioxide. Thirdly it will used in HMP shunt and in their end product will be ribose and NADPH, and which will be used in DNA synthesis, and so glucose is very important for HMP shunt too. It is also used in fatty acid synthesis and glycogenesis in order to make fatty acids and glycogen respectively. So now we'll see that how different tissue in our body use glucose. The brain tissue uses constant amount of glucose for TCA cycles in order to generate ATP, and there will be little glycogen storage into brain cells. In muscles and heart they uses TCA cycles to generate ATP, and the transportation into these cells is heavily influenced by insulin, and glucose stored as glycogen into the cells of muscles and heart. Our red blood cells has no mitochondria, that's why they cannot run TCA cycles, as they use glucose in anaerobic metabolism to make ATP and generates lactate. Also use glucose for HMP shunt to generate NADPH into red blood cells. In adipose tissue, glucose is converted into fatty acids, and this uptake is also influenced by insulin. So now we'll see that how glucose can enter into cells. There are two ways that it can enter into cells. The first is sodium independent entry, that is by transporters. There are 14 different transporters have been described that can carry glucose into the cells, which is GLUT1 to GLUT14, and they varies by tissue. As you can see in the picture that glucose is carrying into cells by GLUT1 transporter. Second way is sodium dependent entry, which is glucose absorbed from low concentration to high concentration from intestinal epithelium and renal tubules that we are going to see in next slides. So here is the picture of glucose transport in kidney. As you can see in the picture that sodium and glucose will enter into cells with the help of SGLT transporter, and that's why glucose concentration in cell will get high in compared to the blood vessels. So glucose will leave the cells with the help of GLUT2 transporter, and high amount of sodium will also be neutralized by sodium potassium pump, and that's how glucose will enter back into the blood vessels. Here's the another picture of glucose transport in GI tract. As you can see that sodium and glucose will get enter into cell with the help of SGLT transporter, and because of that glucose concentration in cell will get high in compared to the blood lumen, and high concentration glucose will go to low concentration glucose independent manner, and that's how glucose is absorbed by intestine. There are certain types of GLUT transporters that you need to know. The first one is GLUT1 transporter, which is insulin independent transporter. It uptakes glucose when there is high level of glucose in the blood vessels. Tissues like brain and RBCs have GLUT1 transporters. GLUT4 transporter, which is insulin dependent transporter. When there is high level of insulin, it uptakes the glucose, and fat tissue and skeletal muscles have GLUT4 transporters. GLUT2 transporter, which is also an insulin dependent transporter, but this is bidirectional enzyme. Because of that it is also used in gluconeogenesis. This is found in intestine, and in intestine it will transport glucose from cells to the portal vein. Also it is found in kidney, liver and pancreas. So thank you everyone for watching this video. Don't forget to like, share and subscribe to my channel, and see you until my next.